We started a project in the lab about 3 years ago that was inspired by this really brilliant paper by Shinya Yamanaka demonstrating that you could reprogram somatic cells back to a pluripotent state with the ectopic expression of 4 transcription factors. And we got inspired, as did the whole stem regenerative medicine community, by this work, which was really brilliant. But there was clear hurdles still standing in the way before iPS cells, the so-called iPS cells, could be translated clinically. And one of the major hurdles was that Yamanaka used retroviruses to reprogram the cells, which permanently integrate into the genome. And we know from the gene therapy trials of the 1980s that you don't want to have viruses in the genome. So we set about the task of trying to do reprogramming in a non-integrative fashion by using another nucleic acid, not DNA, but rather mRNA, which is the intermediate between proteins and DNA. And in order to accomplish that, we had to develop a technology around that platform which allowed us to do it, because it wasn't just as straightforward as it may have seemed to us originally. This project took about 3 years to do from its original inception until we finally succeeded. And all science is expensive to do. We were fortunate enough to have the support of HSCI, which gave us a seed grant, which was designed to fund this very high-risk, high-reward type of project. And I'm happy to say that we succeeded in that to show the benefit of funding high-risk, high-reward projects to junior investigators. The real challenge, once we have pluripotent stem cells, though, is to direct the fate of those cells towards clinically useful cell types, say cardiomyocytes or lung cells or pancreatic beta cells. And this has really been a challenge in the stem cell community. So we applied our technology to this problem and found that we could very efficiently direct the fate of these pluripotent stem cells towards cell types that we could actually use in patients. So I think this aspect was very exciting to a lot of people, and we immediately started collaborating with a lot of fellow Harvard Stem Cell Institute investigators, including Doug Melton, who leads our program, and Ken Chen and Chad Cowan. And we're working now towards generating various cell types depending upon who we're working with. So we're making beta cells with Doug Melton, and we're making cardiomyocytes with Ken Chan, and we're making fat cells with Chad Cowan, and my lab is focused on making hematopoietic stem cells.